Welcome to lecture number 2, Genetic Purity Test for Commercial Hybrids. So before moving in depth into the genetic purity aspects, so first we have to see what are type plants or your standard plants or seeds. So these are plants or seeds that represent the characters of a variety. So this type plants is a representative sample of an entire population. So that is what we have known as type plants or seeds. So next one, what is genetic purity? So it is the percentage of characters showing similarity with that of your type plants or your standard plants. So how much percentage uh, of your characters is showing a similarity with that of your type plants or your seeds is calculated as genetic purity. So here I have given some standardized or generalized minimum standards for your genetic purity for different classes of seeds. So this is only a generalized standards and it can vary. This genetic purity percentage can vary from one crop to another. So normally the breeder seeds are supposed to have some 100% genetic purity, whereas foundation is to have 99 and certified needs to have 98% and hybrids needs to have 95%. So this is the generalized scheme of genetic purity. And there is an obvious relation between genetic purity and our yield component. So it says that 1% drop in genetic purity will result in a loss of 100 kg per hectare yield. So that's much importance we have genetic purity in crop cultivation. So this is the table showing the minimum genetic purity percentage for both foundation and certified seed in various categories of crops. So there are three categories mainly: self-pollinated species, cross-pollinated and often cross-pollinated. So under self-pollinated species, the crops such as wheat, barley, rice, oats, black gram, green gram and chickpea have both foundation and certified seeds at 98% of genetic purity. And when coming to groundnut, it is 96 percentage, and in soybean it is 97 percentage for both categories of seeds. And in case of cross-pollinated species like maize, pearl millet, sunflower, safflower and cauliflower castor, it is at 98 percentage. And for mustard, rapeseed, sugar beet, it is 97 percentage. And for cucurbits we are at 99 percentage. And again for often cross-pollinated species such as cotton, sorghum, pigeon pea, brinjal and chili, it is at 98 percentage for both foundation and certified. And for bendi it is at 99 percentage, and for sesame it is at 97 percentage. So what are the factors affecting genetic purity? So the first one is our natural crossing, and second one is mechanical administrations, the third one is genetic drift, and fourth one mutation, and fifth one is our pests and diseases. So these five factors are the most affecting ones for your genetic purity. So now coming to genetic purity testing, we have three types of testing. So first one is seed testing, and second one is seedling testing, and third one is the field testing. So under seed testing, we have visual mode of testing, chemical testing, biochemical testing, and cytological testing. And under seedlings testing, we go for seedling color and other characters. We will be examining the seedling. And under field testing, we have GOT or our grow out test, where we will be growing the entire variety or your plant in a field and evaluate its morphological traits. So now let us start with seed testing, and under that we will be first seeing our visual testing. So this is the simplest test available, so that's why it is not much reliable. Usually this visual testing is used in combination with other mode of tests like chemical or your biochemical tests. And here the seeds are spread on a purity workboard. So this is your purity workboard. So we have a platform so that you can spread your seeds, and you have a magnifying glass and an overhead lamp over that, so that you can visualize these seeds clearly, and the seeds are tested on the basis of size, shape, and their color. And again, for a better visual analysis of these seeds, we can also use or integrate an UV light into this purity workboard. So it will increase the cost and it is a bit more difficult as we are involving this UV light. So this marks the difference between the seeds more clearly if we add UV light to this purity workboard. And again, the seeds of other varieties distinguishable from the principal variety or your that standard plant or your standard seeds are separated, and number of other varieties that got admixtured into your desirable seeds are calculated. How many other variety seeds are present per kg is also calculated so that we can have an estimate of your GP or your genetic purity. So the second one is chemical testing. So this is used to test the chemical characteristics of seeds. So here we will be using so much of chemicals in different crops to test the chemical characteristics of a seed, and we will be differentiating those seed varieties based on those characteristics. So first one we are going to see is phenol test in wheat. So this is a test used for varietal distinctions where we can distinguish between various varieties. So it is being used for the last 50 years. So this is the picture showing this phenol test. The wheat seeds are suspended in chemical phenol, and it is showing a color differentiation according to the varieties. So by these color variants we can identify different types of varieties in wheat. So this is the experimental setup. So first of all, we have to soak the wheat seeds in for 16 hours in water. So the temperature of the water can be around 20 degrees Celsius. So we need to soak the seeds for 16 hours. Then take the seeds, transfer the seeds onto filter paper in a petri dish that is moistened with 1% phenol solution. And also we can keep the seeds in the solution for 4 hours. So that is the experimental setup. We can adopt this petri dish experimental model also. So now coming to the enzymatic reaction that is happening or that is responsible for the color variation is that in the wheat seed coat we have a chemical called as tyrosinase. So there is the abundant presence of this tyrosinase, and it can vary. This concentration can vary from varieties to varieties, and in the solution we have phenol. So these two chemicals react together to form different colors, four types of color exactly, that is black, dark brown, brown and light brown. Based on the concentration of this tyrosinase, this color variation can happen. So this tyrosinase react with phenol to produce these four types of colors. So Walsh in 1965 proposed this phenol test for cereals such as rice, wheat and jowar or sorghum, and this is very easy, quick and also reliable. And a group of varieties can be identified, whereas the seed of a particular variety cannot be identified. So only a group of varieties can be identified here, whereas the seed of a particular variety cannot be identified. And again, there is a modification for this phenol test where the seeds are soaked in 0.01 to 1 percentage of CuSO4 or your copper sulfate, or Na2CO3, your sodium carbonate, instead of water. Instead of soaking in water, we will be soaking the seeds in copper sulfate and sodium carbonate. So this modified test will give color variance within the group of varieties. So earlier I have said a drawback in phenol test that it cannot identify a particular variety or the varieties which is categorized within a single group. So the phenol test is unable to give the color variation. But once you soak the seeds in this sodium carbonate or copper sulfate, you can get color variance within the group of the varieties. And again, if the seeds are treated with fungicides or those the seeds we are using is pre-treated with chemicals, then it needs to be pre-soaked using methanol. So it needs to be pre-soaked with methanol to remove these chemicals. So next one is NaOH test for wheat. So this is developed by Payne in 1988. So this test differentiates white and red colored wheat seed varieties. So it can only differentiate between white and red colored wheat seed varieties. And again the procedure involves the immersing of these wheat seeds in 5% NaOH solution for 1 hour and observe the seeds after 1 hour. So we need to soak the seeds in 5% NaOH solution for 1 hour. So if the seeds are belonging to this white seed category, after 1 hour, just we can see a dark yellow formation. Just after that 1 hour of soaking, we can see dark yellow, and as the time passes, say some after 10 minutes, we can see an light yellow color formation. So then we can confirm that these are white seed varieties. And coming to the red seed category, so after 1 hour of soaking in 5% NaOH, we can have to wait for 15 minutes. After 15 minutes only, there will be a orange color formation, and again after 1 hour, then we can get a dark orange color. So then after this observation, we can confirm that these are red color seed varieties. Now the third one is KOH test for rice. So this is developed by Rosta in 1975. So here the certain red rice seeds become white and get mixed with our normal white rice seeds. So certain red rice seeds may have become white due to some of the external factors, and it can get mixed with our normal white rice seeds. So in that case, the identification becomes very difficult. Hence, in this particular scenario, the KOH method can be successfully utilized to identify the red rice variety from our normal white rice. So the procedure involves the suspension of the seeds, that is to be tested in a test tube with 2 drops of 2% KOH solution. So we need to suspend the seeds in a test tube which is containing 2 drops of 2% of KOH solution. So this turns the grains of red rice into red, whereas the normal white grains remain unchanged. So now coming to next test, that is Feso4 test for rice. So here the suspected rice seeds are immersed for 4 hours in 1.5% of FeSO4 solution, and on the husk grains we can observe some brown spots and gray streaks, which is resulting from the treatment of the seeds in this FeSO4 solution. So by utilizing these variations, now we can easily identify the varietal admixtures. And next one is our KOH bleach test for sorghum. So it is developed by Payne and Kosikowski in 1980, and the main driving principle behind this chemical test is that the inner layer of the seed, which is known as tegmen, is very much rich in tannic acid. So this tannic acid, when reacted with our KOH bleach, it can cause dark pigmentation. So this is the basic principle involved in this test. And coming to the procedure, the KOH plus NaOCl, that is your sodium hypochlorite 5.25% is mixed together in the ratio 1:5 in weight to volume method. And again, the seeds with the brown coat is soaked in the solution for 10 minutes, whereas the ones with the white seed coat is soaked in the solution for 5 minutes. So we need to visually identify the brown coat and white coat. Then accordingly we have to suspend the seeds in the solution, and the time difference varies. So now the seeds are washed in water and dried on filter paper. Again, the seeds develop dark and light pigmentations which can be used to distinguish between varieties. So now coming to peroxidase test for soybean. In the seed coat of soybean, there is a presence of peroxidase enzymes. So the seed coat of soybeans is able to produce an enzyme called peroxidase. So this peroxidase enzyme is governed by a gene P. So looking into the genetics of this peroxidase enzyme, so a homozygous dominant or a heterozygous alleles are responsible for a higher peroxidase activity. So a cultivar or a genotype of soybean with a homozygous dominant, that is capital P, or a heterozygous combination capital P small p, can produce high peroxidase activity, whereas the homozygous recessive small p is responsible for low peroxidase activity. So this is the genetics behind this peroxidase enzyme that is being produced in the seed coat of soybean. So, Buttery and Buzzell in 1968 developed a method involving the reaction between peroxidase and H2O2 to differentiate between the cultivars of soybean. So now coming to the procedure, the seed coat is removed and placed in individual test tubes. So in each test tube, 10 drops of 0.5 percentage glycol solution is added, and after 10 minutes, 1 drop of 0.1 percentage of H2O2 is added, and if it shows a reddish brown color within a minute, indicates the presence of peroxidase, and if it remains colorless, then we can say the enzyme is absent. So the variations in these reactions means impurity in seeds. So any deviations from these reactions means that there is a impurity in our seed lot. So now coming to the third category in our seed testing, so that is biochemical test. Under that we will be seeing electrophoresis test, and the main principle behind this is that the primary gene products such as proteins and enzymes differ in various crops and their varieties. So this difference between the proteins and enzymes is being utilized to differentiate between the varieties of a crop. So this is very rapid, this is very fast and inexpensive. So that is the major advantage of this electrophoresis test, and in 1993 this protocol was added into the ISTA rules for seed testing. So now coming to the procedure. So the proteins extracted are separated by electrophoresis carried in agar medium, and this will give distinct color bands. So different varieties will give different banding patterns. So thus admixtures of other varieties can be easily detected by comparing the reference banding pattern of the variety. So we can just compare the reference banding pattern with that of the seed varieties that is need to be tested. So again, instead of agar medium nowadays, we are using polyacrylamide gel, or your page is being now used. And again, the conditions of the experiment such as medium, pH can vary according to the variety or the hybrid that is being tested. Next, coming to chromatography test. So this is similar to our electrophoresis test, because here also we can observe different banding patterns like that we have observed in your electrophoresis test. So these variations are based on the presence of flavonoids, phenols, and in certain cases fatty acids. So that is the major difference between chromatography and electrophoresis. Again, the application of these tests are really limited due to its difficulty in execution and long experimental time, and certain varieties tend to show similar banding patterns, so it is not that much reliable. So next one is cytological test. Here the varieties are differentiated on the basis of chromosome structure. So we will be looking for any aberrations there, and we can go for polyploidy level difference in polyploidy, and again for disease resistance, such as we can conduct a pathogen inoculation test so that we can see the extent of the infection of these diseases, and we can differentiate varieties on basis of the cytological differences thus observed. So this is not that much used on a day-to-day basis. And the fourth one is our molecular markers. Here we will be using DNA markers such as RAPD, SSR, and we have ISSR. So these DNA primer based markers are utilized to identify, we can utilize this efficiently to identify the varieties inside a crop species. So after seed testing, now we are going to see the seedling testing. So first and foremost thing what we will be looking in seedling is that its color. So first one is seedling color. So the pigmentation in stem, plumule, etc. may vary from variety to variety. So we can observe some extent of pigmentation from heavy to light pigmentation in the stem and plumule region, and it can vary from variety to variety. So the seedlings can also be treated with UV light or 1% HCl or NaCl. This would enhance the pigmentations and which will make our visual observation much more detectable. And apart from the seedling color, we can go for other characters also, such as nodulations. The number of nodulations can also vary from variety to variety, particularly in the case of legumes. And also these seedlings can be grown in greenhouses to test certain diagnostic morphological traits that are very much unique to the concerned variety. However, the field test is only preferred in crops requiring larger spacing such as cucurbits and tomatoes. So in crops that is requiring larger spacing, seedling testing is not preferred, especially the seedling testing in greenhouses are not preferred. Instead of that, they will be going for field test. So the third category is field testing or your grow out test, though infamously known as grow out test. Here the seeds are sown and a comparative study is carried out. So seeds are grown in two replication and the traits are compared with that of the reference sample or your standard plants. So the observations are made on your seed morphology such as length, width, shape, thickness, etc. by naked eyes or hand lens or your scanning electron microscopes. So then again, seedling morphology such as plant habit, flowering habit, floral characters, fruit characters are also observed in field testing. So now coming to the criteria for GOT, the plant population in the field must be a minimum of 400 plants. At least 400 plants are required to assess the genetic purity using this grow-out test. So other parameters are also listed out in this table. So you can see if the maximum permissible off-type percentage is around 0.10, then the minimum genetic purity percentage is 99.9 percentage and the minimum number of plants required for observation is 4000. And if the maximum permissible off type is 1 and above, then the minimum genetic purity percentage is 99 and below. And again the number of plants required for observation is 400. Now coming to the size of submitted sample for GOT. If we are taking samples from large seed crops such as maize, cotton, etc., then we have to submit a sample of 1000 grams. If it is of medium seeds like wheat, rice, etc., then we need to submit 500 grams. If it is for beet and similar sugar beet and similar sized seeds, then we have to give 250 grams. And if it is of bajra, jute and other similar crops, then we need to submit 100 grams of seeds. So now coming to the conditions of GOT. So the first and foremost condition is that the marker characters may also develop even in off season. So this will ensure that we can conduct GOT or our growth test in the off seasons also. Again, the field should be neat and clean. And again, the standard cultural practices must be adopted. However, the row length and the distance can vary. The examination point of view, the examination or the evaluation is carried out throughout the growing season for all the marker characters, and the plants showing differences in the marker traits are marked and counted, and the genetic purity is calculated accordingly. So that's all about the genetic purity regarding this lecture 2. So hopefully we can see in lecture 3 very soon. Thank you.